Right, thanks very much. Let's see, I will share my screen. I'll go all in and share everything, and hopefully it will work. Can you all see that? Great.
Yeah, we can see.
Let me hide the meeting controls. Right, thanks for that introduction, Michael. And some of you may know Michael and I have been discussing or applying. He's been applying for money to come over to Sweden, and that would be great. So, and he is the reason I've been invited, and with Lena as well, and George to talk to you. I feel slightly unprepared and possibly overwhelmed by the number of proper entomologists in the room at the moment. I'm, as Michael said, a bit of a jack of all trades, but my background is archaeology and geography with a bit of computer science and various things. So I'm partly an ecologist, partly an entomologist, but in no way am I a forensic scientist. So it's really superficial, a lot of this stuff that I'm going to show you. And also I'm drawing on the work of a lot of other people. Small parts of this are my work. As Michael said, I'm also vice dean at the moment, and that means you lose complete lack of, or you gain a complete lack of control of your own time when you do this. So all my planning to run through this presentation carefully in advance and sort out the timing was destroyed by other things. But I will try to stick to an hour, but feel free to wave your hands and shout speed up or slow down, things like that. So I'm going to give you a short overview of palaeoentomology at the beginning, and in sort of looking out checking out your department and seeing what level to put this and what to expect, I discovered Mike's earlier seminar, and he has covered quite a lot of the basics, and especially in terms of funerary archaeology stuff. So I'm not going to talk about any of that really at all, and I think if you weren't at his seminar, have a look at that. I'll give you a couple of sort of forensic archaeology orientated case studies, and those are the Tale of Two Bishops and the Missing Vikings, as I've called it here. And then at the end, I'm going to bombard you with some recent data science work, really, that we've been doing, where we are taking advantage of the large amount of data that has been accumulated on palaeoentomology over the last about 50 years, really, since it started in the 1960s with Peter Osborne and Russell Coop at Birmingham University. And just to let you know where we are, this is Umeå University. Apparently you can fly there in 10 hours. I refuse to believe there's a direct flight from JFK to Umeå. And I know you can get it for cheaper than $1,000, but the Arctic Circle is just above us. It's 400 kilometers, what is that, 250 miles north of us. So we get significant winters, but we also get very warm summers with 24 hours of daylight. So it's a bit of a land of contrast. At the moment it is yellow and all the leaves have fallen off the trees, so we're very much in the fall at the moment. It's a rural environment, mainly woodland around us, and the archaeology here is very much affected by the type of sediments that we have. It's most of northern Sweden is quaternary sediments, so it's post-glacial moraines, very sandy, lots of fine silts, you could say, that don't really allow for good organic preservation. But that's intermixed with peat bogs, where we have fantastic preservation. So we get poor preservation of archaeology from the archaeological sites, but very good environmental records going back to the last ice age and occasionally to the previous interglacial. just a bit of advertising for the university. I have to do that now. It's quite a big university. We're one of the what's called the Big Five of Sweden, but there are really only 9 large universities in the country. There are quite a lot of smaller ones these days. About 36,000 students and just over 4,000 staff. So we collectively make up about 1/5 of the population of Umeå. So it's a student-dominated town. I'm run something called the Environmental Archaeology Lab. And for those of you who don't know what environmental archaeology is, it's all about reconstructing past environments and human activities, environments, climates, and human activities. And what we do is basically look at anything that is preserved under the ground, really. We, at our lab, specialize in looking at organic remains. So here on the left, we have a pollen grain. This is a pine tree pollen. This is a fossil flea from Greenland. This is, oh God, I've forgotten what it was. Looks like, any entomologists in the room can identify that for me, but it's a beetle. And on the right here, we have a carbonized grain seed. And just to the left here, we have a phosphate molecule. We do quite a lot of geochemistry as well. And these things come from lake sediments, peat sediments, and archaeological sites, really, to make a broad sweeping generalization. With the soil chemistry and geophysics, we do a lot of surface mapping to try and identify the areas that have been affected by humans in the past. And the timescales for us really are mainly the last, I'd say mainly the last 6,000 years, going back to 14 or 15,000 years if we're working in the far south of Sweden or in Denmark and Germany, where we've worked before. So we take samples from these environments. I'll show you some pictures of how that works later. And then basically we have a set of different methods. I know you can't see this, those of you who are looking on laptops, but basically we take samples for different purposes, we chop it up into different bits, and then we reduce the samples through different mechanical or chemical treatments to extract the type of material that we want to analyze, basically. And that leads to things like being able to do distribution maps for certain species of beetle, in this case Bryophyllus hiningianus, and this is extracting data from the database where each one of these sites where this species is found has contributed to us to being able to map it, with these green dots here showing the place but not the time, of course. The grey shaded area here is the modern distribution of this species, so you can see it was present in much colder climates throughout the British Isles and other parts of Central Europe. We do quantitative environmental reconstructions, and I'll come back to that. It's quite important for the way we work. We do climate reconstructions. I've done this using beetles before, but there are various other methods. One thing we started to do now is to get involved in cultural heritage conservation, and in particular trying to predict where archaeological sites and environmental deposits are going to be at risk to damage from future climate change. This is, as you all probably know, an accelerating problem as global warming accelerates. What you can see here is some of the surface mapping that we would do. This is a soil chemistry map, and the increasing blue colors are increasing levels of phosphate, and these are two Iron Age buildings from the archaeology, with some little dots showing where the macrofossils have come from the post holes that supported the roof of this house. And then a very nice pretty picture that is a final interpretation of it. And quite often we follow this chain from analysis through quantitative reconstructions to a sort of narrative, often pictorial picture of what we think happened there. We deal quite a lot with multiple scenarios in this thing as well. There's never one clear picture of what has happened in the past. The evidence can point in many ways. So fieldwork, This is a picture of the equipment storage room in our lab, one of them, a bit of a mess. We grab things out of this and then we head off to different parts of the world to take samples. Here is a bit of the, I can call it geochemistry, but it's on a castle wall in France, doing XRF, X-ray diffraction, and near-infrared spectroscopy there to try and look at the origins of the building stones and the mortars that made the Carcassonne castle. This picture here is using a near-infrared probe to look at and try and understand the pigments that were used for Mesolithic rock art in the middle of Sweden. And then we have sampling in Iceland in a medieval Viking Age farm site that is basically covered by volcanic ash and sand. In the middle here is desperately trying to get samples from a paleo channel, so an old river in eastern England before the old river came back to the big hole we dug. We work with drones, of course, for doing landscape mapping. We use a lot of LiDAR data, laser-based terrain data, and we do a variety of coring techniques. This is a Russian auger pushing down into peat bog to bring up cylinders of sediment. It's quite muddy work, you could say. These samples come back to the lab, and this is pretty much standard for any type of lab. We have various ways of processing, reducing the samples. We will burn some of the samples or heat them. If we're looking for carbonized seeds, we want to get rid of all the moisture so that the samples don't go moldy, really, and we can extract the charcoal. Chemical treatment, so if you're looking at pollen, we need to get rid of all the minerals and everything that isn't pollen, really, so we use some quite nasty acids to break down the silica there. And then a whole series of analysis methods. Spectroscopy, photospectrometer is really our workhorse for looking for things like phosphates. And the most interesting bit for me really is the microscope room, where most of the insect work is done. And one thing I want to stress here is the importance of retaining data and metadata from all these, well, all the aspects of the analysis, so that anybody trying to reuse the results of our analysis later can understand where they come from and what the potential limitations are of reusing that data. And I'll come back to that when I start talking about databases later. This again is a plug for a project that we just got funded last week, so I'm still very excited about it. We have got funding with 9 partners throughout the country to build a national infrastructure for digital archaeology, and our lab is going to be responsible for the incorporation of scientific analysis data into that system. And that's building on a database we have called SEED, the Strategic Environmental Archaeology Database. And the idea there is to collect all the data from all forms of archaeological analysis, even quaternary science analysis, and put it into one place, make it openly accessible, and allow lots of really cool things to be done. I'll show you some of the cool things later. Why? Really, and that's a massive question. The past, a lot of things have happened in the past, but what we can see of this is filtered by a whole series of what we call taphonomic processes. The main one of these is loss of evidence. So not everything gets deposited into the kind of sediments that preserve it. In fact, it's a minuscule proportion of what has happened in the past that's recorded in archaeology and geology. A lot of sediments get eroded away, a lot of things decay, and increasingly so human activity destroys these evidence. We have peat extraction, development, and draining of peatlands, building of roads, building of railways. Pretty much everything humans do will damage some form of evidence on what's happened in the past. The other thing that is becoming more and more poignant, you could say, is that even if we are recovering a lot of this evidence of the past, we are not often saving the data in a way that can be recovered for future research. So there's both a physical and a digital filter, you could say. This thing that's reducing here really is the picture of the past getting constrained, and then we have to try and extend it again or expand it to build snapshots of what's happened in the past. And you probably all know the old adage, the past is the key to the future. And really what we like to say here is that you can't possibly predict what's going to happen in the future without understanding what has happened in the past. So we're building the basis for any model of what's going to happen in the future. And that's natural processes to human behavior, really. What we like to say now is that archaeology, every other form of science, is a support discipline for archaeology. We gather everything. It's a bit pretentious, but we like it that way. So on to palaeoentomology or archaeoentomology. The terms are pretty much synonymous, I would say, though some people maybe prefer one term to the other. If you work more on deposits from archaeological sites and more in terms of human impact, human activity, then archaeoentomology may be your preferred term. Palaeoentomology is either more orientated towards natural environmental change, natural deposits, large-scale landscape change, but really the two merge. Once you get 2,000 to 6,000 years ago, and depending on where you are, it's very difficult to separate out the two. And you can't understand what humans have done without understanding the natural background anyway. We mainly deal with what is called in some places subfossil material, especially in Scandinavia, it's called subfossil. But really, the definition of fossil means anything that you dig up from under the ground. So the term fossil does apply for this. But subfossil is used to basically say these are not mineralized, generally not mineralized fossils. And they are quite recent. The recent fossil record is another term that's used. Palaeoentomology, from my perspective, is really quaternary entomology. So we deal with the last 2.6 million years. That's flexible. Some of the oldest fossil or the oldest non-mineralized fossils come from Antarctica and are about 4 million years. That's Alan Ashworth's work from North Dakota State University. Most of the work is really over the last 13,000 years, so since the last ice age. And there are sort of clusters of samples or clusters of papers based around certain individuals. This is, the shading here is the peak according to Russell Coop, who is one of the founders of palaeoentomology, or at least modern palaeoentomology. He really liked the late glacial and trying to understand the rapid climate change at the end of the last ice age and the beginning of the Holocene. If we look, and these are radiocarbon dated samples, so this isn't the entire record, and this is a few years out of date now as well. If we look at the more archaeologically orientated data or work, the Roman European, or the Roman British, the Roman Northern European period is heavily researched with palaeoentomology. And then the last 1,000 years, again, is really heavily investigated. And of course, if you're looking at long-term environmental change or long-term studies of insect biodiversity, you're going to have to look out for these variations in the density and quality of the data, because your biodiversity statistics are going to have to be standardized really to compensate for any of these problems. And also the selectivity of archaeologists is very important to remember there. It's a biased record. We're looking for human activity generally. So sampling. As I said, many different approaches. Here we have a core from a Russian peat corer, and you can see the different degrees of fumification, so breakdown of organic materials in this, and also possibly some evidence of burning of the bog or burning of the environment by the injection of charcoal into different areas of the sediment as it accumulates. Most of our sampling is looking down holes and trying to extract lumps of peat, which we put in bags. We tend to work with 5-litre bags. Sometimes we will extract what we call a monolith, so a lump of peat, and then we'll chop it up in the lab. And that allows us to do things like pollen analysis, where you need very, very small samples from the same core and then correlate the evidence from the different ways, the different types of data or the different types of organisms. We do geochemistry of the core as well. And that's very important because the different methods complement each other in different ways. Every type of evidence has its own limits, you could say. We float that, so we wash out any fine mineral material. So we use 300 micron sieves generally. That's standard practice for fossil insects, but if somebody wants to get out smaller seeds, then maybe a 250 micron will be used. And then if there is a lot of organic material, we will use paraffin flotation or kerosene, I think it's called in the US, because that bonds to the chitin and makes the insect remains preferentially float over the plant's macrofossils. And finally, we end up with something looking like this grey goo in the sieve. We then sort through this goo and under a binocular microscope and pull out anything that we think we can identify. Most of this is Coleoptera, so beetles, but also there is a lot of work done on chironomids from lake sediments, so this is larvae, Trichoptera, caddisflies, also from aquatic sediments, the larvae. A reasonable amount of work looking at Diptera, but it's mainly only the pupae that survive. And Orobatid mites is getting a bit of a resurgence now. But if we find something interesting, we will try really hard to identify it. And after picking everything out, it looks like that. If you're lucky, it can be damaged. You can get a lot of variable preservation, but what you can see is the parts of fossil insects. So here you can see a carabid in the middle there and another one there. Here's a bit of a water beetle. Sorry, these are Elytra. There's a staphylionid, rove beetle. I don't know, maybe an Aphodius, a dung beetle there. We've got a couple of rest eggs from water fleas, Daphne, there. And we basically compare these to reference specimens. We use a lot of images on the internet these days. There's some great picture databases out there. And if something is tricky or we want to be extra careful in our determination, then we work through standard entomological identification keys, quite often a bit backwards. We start with what we think it is and then go back up the analysis tree to figure out whether it works or whether it doesn't work. So it's a bit of a puzzle, it's a bit of a game. It's fun for a while, but after a while, if something is very difficult to identify, it gets frustrating. Here you can see a few fossil specimens that are still partially articulated, and that's quite rare. That's an indication of good preservation. That's a large weevil head, actually, upside down, which I was trying to figure out what the big circular thing was when I was looking at this earlier. How much data do we have on these things? We have a database called Bugs Coleopteran Ecology Package that is ingested into seed at the moment. There's a bit of a lag there. I'll give you some web addresses later. But we have almost 1,400 sites. And about half of those are archaeological, half of them are natural deposits. There are other databases out there, the paleobiology database, that includes much, much older fossils. I think they stop at about 1 million years ago. Neotoma overlaps seed, and the, now what's this called? The quaternary insect database of Siberia is sort of geographically isolated from us and unfortunately not publicly available, which is a bit of an issue. All in all, this is over 750,000 fossils that have been identified through this somewhat time-consuming process. I think once I calculated how much time that is, but it's quite a lot of years of work through many, about 2,000 papers over the past 50 years. We have just over 5,000 taxa identified in these fossils, and 3,700 of those are to species level. So for fossils, this is very good taxonomic resolution and allows us to do very detailed environmental reconstructions. There's the data from paleobiology database for the Coleoptera and Neotoma. We work a lot with each other in terms of data exchange, which is almost working. So when we have identified our beetles from our fossil samples, we want to know what it means. And the first things we do is figure out, where does this species live now? We look for earlier records of it as a fossil to try and understand how it's moved over space and time, or been moved over space and time. And then we look at the ecology or the habitat, the food sources of each species. And we look at the climate range of that species, if it is a species that is constrained by climate. And of course, a lot of these things are debatable. Like any form of science, there are a number of assumptions that we make to enable this to work. And these are assumptions that apply to paleoecology in general. So one is that we can identify the fossils using modern specimens. And this is something that becomes more and more discussed with the advent of DNA work for defining what a species is. Eventually the DNA people will agree with us, but we can maybe talk about that later. Another assumption is that the habitat preferences from the beetles' point of view have not changed over the period of the 2 million years that we're interested in. And that is, of course, open to debate. A lot of our reasoning can be considered circular. And if we were just looking at fossil insects, this would be a serious problem. But the fact that we also look at seeds, pollen, and a lot of other methods to corroborate the interpretations means that we're pretty confident that this assumption holds for most species. But of course, it's not going to hold for every species. And that's why we never say our interpretations are 100% reliable. They're always open to reinterpretation. And the same goes for climate tolerances. We assume that the beetles migrate as a population, that they can rapidly move as the climate changes, rather than evolving. And of course, beetles and insects have been around for a very long time, and you could somewhat, I don't know what the word is, but we could say that they've done all their evolving, so maybe they don't need to do it so much at the moment, at the present time. But also that's, of course, open to debate. So the visualization of that process. So here we have a part of a fossil species list. This is from some samples from pits in eastern England from the Bronze Age, so about 4,000 to 6,000 years old. We look at the existing fossil distribution for those species, and then we pull up the ecological data from as many sources as we can find, really, or can reasonably find, given the time constraints for each of those species. And we summarize those, or we have our own systems, our own, you could say, archaeology-orientated quantitative classification systems for the species, which allows us to do some form of quantitative environmental reconstruction. But also we use a lot of the descriptive data there. And then we build a narrative of what it means. This particular example, I forgot to put up the reference, we reinterpreted some archaeological evidence of waterholes to say this couldn't possibly be a waterhole because it was full of dung, which is quite fun when you can turn over some established interpretations. So the forensics examples, or forensics orientated examples, if any of you have been looking at paleoentomological literature, you'll see two names popping up quite a lot. One is Eva Panagiotakapoulou, and one is Paul C. Buckland, who is my father, I have to admit. So most of the work you see published with Buckland, P. Buckland, is actually his rather than mine. He brainwashed me at some early age. I tried desperately to do different things as a teenager, but failed abysmally. Excuse me. So this is a slightly, forgot the date. This is about 2015, 2016, a study of a buried bishop at York Minster in England. And then the other more recent study that I was actually involved in is a study of 17th century bishop from Lund. And that was just a year or two ago that we wrote that. So two bishops, similar functions, similar positions, about 300 years between them. We know reasonable amount about some of them. We don't know when Bishop Greenfield was born, but we do know that he died in 1315 and that he was the Archbishop of York, which at that point was the capital of England from 1304 to 1315. Bishop Winstrop, we have a lot more information about, naturally, because it's a more recent. He was, interestingly, the Danish Bishop of Lund until the Swedes took it over and he became the Swedish bishop, and he very cleverly changed sides at that point. If we look at Bishop Greenfield first, we have some historical records, both in terms of the burial. He has a very, very impressive tomb in York Minster, which I recommend visiting if you ever get the chance. He was in what was supposed to be a sealed lead-lined coffin, and this was quite common in this period, that important people, you sealed them in to preserve them, basically. In 1735, the tomb was opened, and I can't remember what the ring was about, but it was quite important, and it's now on display in the museum. And in 1973 or something, they opened the coffin and took a sample of pretty yucky, smelly stuff. That was processed using the standard paleoentomological method. And what my father got out of this was a small assemblage of mainly beetles. So I'll tell you a bit more about these in a bit, but here we have the staphylinid head of Quidius mesomelinus here, thorax, and an elytron. So all the bits, and we make basically a puzzle like this down here, where we try to fit all these bits together to get a more reliable identification. A Rhizophagus parallelicollis, let's see, there we have a thorax, and Mycetea subterranea, there we have an elytron. And excuse me, this isn't COVID, I'm just I'm not talking so much when I'm not lecturing these days so much. We got, or they got, quite a lot of fragments of coffin flies out of this, which are forensically important species, but unfortunately not identifiable to species level, so the interpretation is pretty vague there. But the graveyard beetle, or the coffin beetle, the Rhizophagus there, is very, very useful. And the Quidius is a predator. It's quite common in archaeological deposits. You get it in waste heaps, in floor layers, and it's basically eating a lot of other stuff. It's a secondary predator. In summary, this is a post-burial fauna, and these are all later corpse colonizers. So it's not the first, it's not a body that's been exposed and colonized by a standard, what you would say, early stage decay fauna. But our interpretation there is that the coffin didn't seal very well, so he wasn't protected from these creatures. Moving quickly on to Bishop Winstrop. He was considerably better preserved. If you don't know of this case, then I recommend Googling it, or I can send you some references, because there's some fantastic CT scans of his coffin and of the body, and you can do a 3D exploration of this. He was laid on a bed of plants with fully clothed. He was put in 2 different coffins, and we think, and the historical documentation seems to suggest, that he was stored for some time before he was buried properly. And this isn't unusual for revered religious figures. He was he's a mummy basically, but he was not embalmed. There's no evidence of that. So the body suggests that he was stored in a cold, dry place. Christopher Fagerström at Lund's Biology Museum took a lot of samples from this and worked very, very thoroughly, painstakingly through it, and got a considerably larger list of species, including over 600 Typnus unicolor, and this is a spider beta, sorry, it's a Paulus unicolor in the Swedish taxonomy. A spider beetle, which is quite common in dark places, basically. My barn is full of them. There are quite a few interesting things in this. We have some mites, and there is evidence of mites biting the hands of the body inside the gloves. And one of the things that is discussed here is how much of these things are introduced during different opening phases in the coffin, and how much is the original stuff that comes in from the material and the body before it was sealed. We also have evidence of plants through aphids and evidence of parasitism of the aphids. So it's quite a nice bit of ecology within this body, or this coffin. Some of this is confirming what we know from the historical record, but again, not a fauna that you would expect from a decaying body. This is a special case. And so I think these, from a forensics point of view, these special cases are quite interesting. We in archaeology maybe do not look for the forensic evidence that you would look for in a contemporary forensics case, but there's certainly a lot to learn from the different subjects. Yeah, I was trying to summarize that the 2 dead bishops are not the equivalent of a dead and decaying rat, which my very supportive wife allowed me to keep decaying in the garden over a summer and take a whole series of photos with our usual necrophorus species and things like that, and all the calophorids eating it away there. It's pretty smelly, but that's not what these guys were. As an environmental archaeologist, I always like to do a quantitative environmental reconstruction from everything. It's not necessarily valid, you could say, in terms of the depositional environment, and the way we have classified our species may lead to some very strange things. But always, this kind of visualization, I think, helps in the interpretation of any kind of data. And one interesting thing there is the ectoparasites there. So Christopher found Sweden's oldest common bedbug fossil, and really we should have made more of a thing of this, since bedbugs are so popular or unpopular at this moment. And I found on your seminar series a very interesting presentation about bedbugs, so that was fun. If we look at the fossil record of the bedbugs, our oldest known fossil record is from Egypt, and this is Eva Panagiotopoulou's work mainly. And this is from El Amon Aranifa's house down here in Egypt, and this is from the database again. And then we have a whole series of more modern records through, well, this is from Quebec, and then quite a few in the UK. Ranifa's house, or sorry, the site in Egypt, is one extreme of the type of environment that preserves fossil insects well. It's a desert, completely overrun by sand dunes, and any fossil specimens are desiccated. So they are extremely fragile, and if those sediments get moved or sorted or processed as we would a peat sediment, for example, they would be completely destroyed. So here Eva was sitting down picking out these things very, very carefully. These are fly puparia, by the way. And there is a paper on DNA in these. But the DNA from the beetles didn't work, unfortunately. And there is the fossil bedbug abdomen. When we're dealing with fossils, we're dealing quite often with different levels of taxonomic resolution based on how well we can identify things. So this is definitely the common bedbug, whereas we have other fossils that are just identified to the genus Cimex. And so if we look at the sites, if we go up a taxonomic level, we get a few more sites, so which probably are the common bedbug, but we're not entirely sure. And this, of course, is quite important in terms of looking at historical trade and the movements of people and pests over time. If we go up another taxonomic level to the family, we get even more sites. And what we can see is that, at least for the British Isles, most of these records are coming in through Roman Britain, and then you have another peak in the medieval period. And this is probably really representing the movement of people and density of populations and the movement of products, rather than any bias in the archaeological record. And Eva has done quite a lot of work on this. If there are any Monty Python fans out there, you have the famous "What have the Romans ever done for us?" And one of the things is that they brought us a lot of pests, including Cytophilus granarius, the grain weevil, which is one of the main pests of stored products to this day. And Eva's work can see basically that species being moved through the British Isles with the Roman army, really, and their massive grain stores, which was something new to that to the British Isles that's never been seen before. She also has looked at this in with my father, a longer time period and a larger geographical range, and can see that quite a lot of these pests or stored products do come with the introduction of agriculture. And they move from the Near East, basically across Europe. This is 9,000 years ago, sorry, moving across the continent. And here we have some of the oldest fossils. This is unfortunately unpublished, I think, from Eva's PhD work, and these are very, very well preserved carbonized grain weevils from a granary. And here is from the same site I showed you before, Amana, Ranifa's house there, a Cytophilus thorax. So I recommend these papers if you want to look at this more. Another example. So the late Peter Skidmore was part of I would say, a generation that's disappearing now as a real, I can't remember the word, an old school natural scientist, really. So he was an artist. These are his paintings, which are amazing. And he was an ecologist and an entomologist, worked at Doncaster Museum. He is most known for his work on diptera, flies, and dung, basically. The world expert on it. And that led him to do a PhD quite late in his life, where he worked on material from Greenland and, to an extent, Iceland. These are fly puparia from a Viking Age or medieval site in Greenland called the Farm Under the Sand. And you get the other end, you could say, of the preservation, as good preservation as a desiccated sample from Egypt, but in this case we're talking frozen. Everything in between those two extremes is worse, you could say. When we are sampling a site that has such potential for interpretation, we tend to do it by room. So we want to know what is the function of the room, and we do it over time. So a room can change function over time, of course, but we also want to know what is happening on this site over time, from its establishment to its abandonment in this case. And the samples tend to look like this. So the Vikings were messy, you could say, but this is intentionally messy. You let things fall on the floor, you cover it with bits of wood or chips or grass or branches, and all the rubbish accumulates, gets compacted, and then when the roof gets too close to you, you clear it out and throw it out into the waste pit, and then use that waste pit to manure your fields. And areas like the corners or the edges can preserve a pretty good continuity of this, but of course you're going to get hiatuses where bits are missing. And we try to understand that in terms of where the samples come from, and this sort of cyclic process of bringing in things from outdoors, throwing them out again, throwing them on the fields, and then bringing them in again. And it's quite fun. It's a puzzle putting all of this together, and then arguing with other people with different types of evidence about what it really means. Sometimes it's easy. So this is the skeleton of a goat, or the skeleton and the wool and even the stomach of a goat. Our narrative is that this is the goat that came back to the farm after everybody had left and wondered where they all were, and then just fell asleep next to the wall and the farm collapsed on it. Unfortunately, the goat is a bit older than that, so the narrative doesn't work, but it was a nice story. Sometimes they're more difficult to interpret, so this is a sort of gooey floor layer. And if we look at the site Nipiatsok, on the western Viking settlement of Greenland, which looks like that, the present day, we've got a lot of different types of evidence, and I'll look at two of these. This is the work of quite a lot of people over quite a lot of years. I wasn't at the site. I was at Gus, there, just across the other side of the river, which is a different but also interesting site. There's a lot of bone, and there's a lot of different bone in different rooms. There's also changes in the type and amount of bone over time. And as you go up in the layers, we get more smaller animals, whereas more larger animals in the beginning, including more domestic animals at the beginning. Pete looked at the flies, and what he could say was these layers are when this site was occupied, basically because there are flies which require a warm temperature. These are fly species that could not survive outside of a human warmed environment in Greenland. They would be killed off by the winter. So these are things that are living their full life cycle in the human-made, the anthropogenic environment. But we have other rooms where there is a cold fly fauna, and so with the bone evidence we can interpret this as a storage room. And then we have all the sort of eating, decaying bodies and things outside, where basically they're throwing all the waste out of the door. These are calophorids mainly. If you keep an eye on what happens to this bit of graphics when you go up in the layers, so these are the final layers, we can see that these species then suddenly, this is happening maybe over, yeah, I guess it is suddenly. These species are spreading throughout the house, and this means that the humans are no longer there heating the environment. They've gone away, basically. You can also see that the calophorids come in and start spending parts of their life cycle within this environment. So here we can maybe assume the door is gone, but also people are not keeping a living environment there. There's decaying things, basically. So this is abandonment. The people have gone. Back to the bones, one thing to look at here is, or think about, is why do they change their diet? And there have been a lot of attempts to explain this and a lot of parallel theories. And one really interesting thing is they definitely eat the dog. And if you're in a place where you need a dog for hunting, you don't do that unless you have no other choice. We were hoping to find bodies, but we didn't. And there are no graveyards, there are no graves or human bodies from the Viking Age in Greenland, which is interesting. And this is a horrible slide with far too much information, but basically it's showing you there are multiple explanations for the collapse of the Viking Age or the Norse colonies in Greenland. And a few of these we can eliminate on the basis of the entomological evidence and the different bits of archaeological evidence together. So I see time is running out here. Do I have another 8 minutes? Is there anybody in a position of authority to tell me to stop, continue, or should I just keep going?
You have 8 minutes, no problem.
Okay, great. So I warned you here, I'm going to bombard you with things on recent work at the end here. And one thing I wanted to show you was how we are working to try and connect the fossil biodiversity data to the modern biodiversity data. And this is through our database, SEED. And the bugs database is what we call a virtual constituent database within that system. So it exists as its own entity, but its data is in that database now. And a other Swedish project called the Swedish Biodiversity Data Infrastructure, which is connected up to the Global Biodiversity Information Facility and part of the ecology network of data providers, you could say. So we're working on connecting these, and it's interesting, but it will happen. So SEED, if you go to browser.seed.se, you can explore all this data. And really, this is an interface for exploring the data in an aim to seeing whether there's anything interesting or useful to you, and then you burrow down and extract it. Unfortunately, the extraction tools are not really working at the moment, but there is an API, which if anyone's interested, give me an email and I'll send you a link. It's what we call a faceted browser. So the smallest divisible element in the database is the analysis of one thing from one sample at one place and at one point in time. And that allows you a very, very detailed granularity. So if we were to filter for all records of Cytophilus grenarius, it will give us all of these dots on the map. We can then tell it, well, we want to know what's the ecological meaning of this. We can ask for the bugs ecocode code system, and then it'll tell us this is a synanthropic species associated with people, and it's a pest of stored grain. And then it will give us a list of the sites. Because of the granularity in the database, we can flip that round, say, now we just want fossil beetle data. We want to use this ecocode system, and then we want to know which species are stored grain pests and where they've been found. So now you can see Cytophilus grenarius is just one of many records and many more dots. So it allows you to look for something, then see what that means, and then take it one step further. And the idea here is to connect that through to the modern biodiversity data. Looking at an individual site from this database, you can go in, visualize it, and export all that data. It's quite standard database stuff, but it's quite new, really, to have this amount of open data available for paleoecology. The reason for doing things like this is looking at one site, reusing that data. That's a sort of repository function, and that's fine, but it's much more powerful if you can pull out data from multiple sites and start comparing, say, your new sample to the data from many, many other sites. And this is some work we did looking at one sample from a Roman well in Sherwood Forest, actually. And we could see that Sherwood Forest wasn't very wooded, which upsets quite a few people. but I'm afraid Robin Hood didn't run around amongst the trees. There weren't that many trees there. And then we compared it to all the other Roman wells in that period, in that geographical area. And then we compared it to all the Bronze Age, so all the wells. And we can see that there are some differences and there are some similarities. And these wells probably are not wells, they are water holes. And some of them aren't even that, the Dung example. This Wilsford waterhole well is something different. It clusters with the Roman ones. And think of wells as giant pitfall traps, basically. And then we can do lots of fun statistics of these things. We can sort of look at biodiversity indicators. We can do climate reconstructions. This on the right is climate reconstruction of all of the sites from the British Isles from the last ice age, and trying to understand what's happening in terms of temperature variation during the ice age. Are we just getting the warmer bits preserved when it comes to the environmental evidence? And of course, comparing, this is some of Russell Coop's work, comparing changes in temperature to changes in environment. The next way to do that is take that up to continental scale. And this is some of a research assistant of mine, Francesca Pilotto, building on Geoffrey Lemdahl's earlier work to redo the climate reconstructions from the 90s with a lot more data. This is also Francesca's work looking at the fossil record of endangered species. So here we looked at the fossil record of species that are threatened in Sweden and tried to see what can the fossil record from the rest of Europe contribute to our understanding of why these species are at risk at the present day. And this is more work with Francesca that we're trying to do sort of a bit of shotgun biodiversity statistics for the entire fossil record of beetles, looking at things like diversity indicators, changes in the numbers of individuals and the number of species, and the environments represented by them over time. This is 20,000 years ago. This is the present day. And trying to understand what part of that diversity change is caused by humans, what is a reflection of variability in the sampling, and things like that. We think humans come in and do agriculture there and affect beetle biodiversity at that point. And we've been doing some network modeling to try and prove that statistically in a way that isn't dependent on the sampling and the resolution of the dating. And this is work through Alexis Royas Vicento, who is a network modeling person. This one is been rejected by several journals, but is on the way down the trail and hopefully published soon. It's interesting stuff. And my final point is the next stage of this. That was just looking at the beetles with a bit of vegetation thrown in there. But when you make the data openly available like this and machine readable, that allows you to connect it to a massive number of other similar systems for different purposes online. And one thing that's quite interesting to do there is not just link the data, but to link the interpretation or the meaning of the data. So for the biodiversity links there, Cytophilus granarius is linked to Cytophilus granarius from the fossil data to the modern data. It's the species data being linked. But in a project called Data Arc, what we played around with was linking things like stored grain pest to archaeological databases, to saga databases, and to tephrochronology databases, and to historical records and things like that. So we're linking the meaning and building a semantic network out of it. And that leads to the potential for very different ways of understanding the data and working interdisciplinary. So I've finished exactly on time. So thank you for listening. And I apologize for the massive bombardment at the end there. I didn't time that very well.
Do we have any questions?
Let's see.
If you have a question, go ahead and unmute yourself.
So I have a question. This is Rick Ludischer. And excellent talk. I have a question about the use of spectroscopic imaging and whether there is any applications. I know that some insects fluoresce. They may phosphoresce, I do not know. I'm wondering if anyone has ever used any digital imaging, luminescence imaging to identify any of these fossils.
The quick answer is no. Not that no, the quick answer is not that I know of, but we have been discussing it. There's nothing published on it that I know for looking at the fossils. My colleague, Johan Linderholm, works with hyperspectral imaging and near infrared and Raman and XRF and things like that. And he is quite insistent that we can use near infrared to identify beetles. I'm skeptical, so I said get a grant and prove it. So it could be worth doing. What he thinks we can also do is scan peat profiles and look for areas of the sediment which have a higher density of fossil beetles, and so it would be a good place to start doing the analysis and things like that. So yes, good idea. Please try it, I think is maybe. One thing to think about there is if we use paraffin flotation, I don't know how that's going to affect the possibilities for analyzing the data. Sorry for using that kind of imaging. It certainly messes up any possibility of doing radiocarbon dating at the moment. So something is happening to the chitin with that. Well, it's bonding with the chitin basically, and that's why they float, of course, when we're doing paraffin separation. The other thing is we tend to store our specimens in alcohol or ethanol, and so that is
that
probably
would probably pull out a lot of the chromophores that might be of interest. Yeah.
So I mean, if we were to do something like that, we would need to either dig out samples that have come from samples that have not been treated in that way, or take some new samples, basically.
Thank you.
Do we have any other questions? Okay, hearing none, I guess we'll bring the seminar phase of this to a close. I thank everyone for joining us. And those of you in the seminar class, please stick around. Philip will take a 5-minute or so break to if you need to do whatever you might need to do. And we'll come back and we'll use the same link that we're using now. Okay, so we'll all be back in about 5 minutes.
Okay, see you soon.
Thank you for the excellent seminar. It was really interesting.
Thank you, Elena. It's good to hear.
Yeah, thank you. Thanks again for coming. Really appreciate it.